Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Today we are going to discuss about cell migration. So what is cell migration? So as the name itself indicates, cell is moving from one place to another place or from point A to point B. So that is called as cell migration, as simple as that, right? So in order to understand how exactly cell migrates inside our human body or any living cells, okay, that we will have to study this topic. So cell migration plays an important role in all physiological as well as some pathological processes. Both physiological and pathological processes, the cell migration plays a very important role that has to be, that has to undergo the cell migration. Then only some physiological aspects and the pathological processes will occur. Without the cell migration, they will not. So that's why they play a very important, the cell migration plays very important role. Let's understand how the cell migration happens in this topic. So cell migration is very important during the organogenesis and embryonic development. So here two things are mentioned which are very important aspects of our particular physiological and pathological processes. We are having the organogenesis and embryonic development. So it plays a role in tissue repair also, a response during the both wound healing and angiogenesis. Even if you are talking about the physiological changes or even in any other kind of like blood any new cell formation like wound healing and angiogenesis, like where blood cells are formed or blood vessels or vesicles or the blood tubes are formed, there also the cell migration plays a very important role. So on the one hand, the immune system relies on the cell migration because the immune system also requires the cell migration to happen, while other hand the cancer metastasis is characterized by the cell motility. So even the cancer metastasis, that is the different phases of the cancers, that is also characterized by the cell motility, how efficiently, how effectively, or how much this particular cell has been moved, okay, or proliferated or differentiated. All these things come under cell movement. Okay, now the above mentioned are the ones that are normally happen in the body. So that happens in the body most of the times, but there are cases where migration of this cell is induced by infusing a chemotactic growth factors. So sometimes this immune system relies on the cell migration, and even the cancer metastasis also characterized by cell motility that is happening in naturally. But in some cases what happens, we need to induce sometimes the situations come in such a way that the cell motility or cell migration is induced in certain cases. How they induce? By using or by infusing, by giving the growth factors. So for example, bone marrow stem cell that can be induced to migrate out of the bone marrow into the blood circulations by giving the chemotactic growth factors into the patients, or by infusing chemotactic growth factors into the patient. That means here we are taking the bone marrow stem cells out of the bone marrow, and these stem cells are going through the into the blood circulation, so that we can take it out for the for example, usually for vaccine preparation, all those things and all we use. Okay, so that's how the things works. Okay, so here basically, and what is understanding the cell motility or cell mobility or cell movement is migration is important. That's it. Okay, so based on the stimuli, there are 5 different modes of cell migration based on what exactly the stimulizing factor. So for example, we are having the chemotaxis, hepatotaxis, galvanotaxis, contact guidance and contact inhibition. Let's say what are the stimuli. For chemotaxis, we are having soluble concentration gradient. This will act as if there is a change in the soluble concentration gradient, this particular stimuli, the cell based on the cell mobility or cell migration happens based on the soluble concentration gradient. Then we are having the hepatotaxis, that is if it is insoluble concentration gradient. Please mark these differences. Chemotaxis for soluble concentration gradient, hepatotaxis for insoluble concentration gradient. Do not get confused. Next, galvanotaxis, the cell migration occurs based on the electric currents. Whereas contact guidance migration happens based on the surface topology. Contact inhibition, that happens based on the lamellipodium, that is cytoskeletal protein actin, which is inhibited by the neighbor adjacency. So these are the 5 different stimuli where the 5 different modes of cell migration can be seen. We are having the chemotaxis, haptotaxis, galvanotaxis, contact guidance, contact inhibition. So cell migration is a 5-step process. Okay, again coming to the next 5s. Now the migration of the cell over a substrata is a fundamental and as well as critical function that requires coordination of several processes or several cellular processes which operate in a cycle. That means obviously for a cell migration, we cannot say that just a cell has to move from A to B, but in order the cell to move from A to B, the other cellular processes also involved, that they also play a very important role, and they have to that coordination should be there. So at the level of light microscopy, the cycle can be divided into 5 steps. That means at the level of light microscopy, in the sense at that particular observation from the light microscopy, we can able to differentiate the cell migration at different stages. Okay, we know that the cell, how it migrates. We can view the cells under microscope, right? So with the help of light microscopy, they have categorized 5 different steps where how exactly there is a significant difference in their movement of cells. So the first and foremost step is the extension of leading edge. Now whatever the leading edge will be there, now imagine this is a cell, whatever the leading edge will be there, that will extend like this, it will extend. That is first step. Because as the cell grow, as the growth factors are added, these particular things, the cell will start extending like this. I hope you are seeing. So adhesion to the matrix. Next topic is it has to adhere in the sense it has to attach to the matrix contacts. That means whatever the substrate is there, that will be a matrix contact. Now the contraction of cytoplasm, because of this, now extended, now it is attached. Now the contraction of cytoplasm will be there. So the cell will become something like this. Okay, because this cytoplasm contracted. So once this becomes like this, next release from the contact sites. Now here from the back end or rear end, it will release like this, or from here also it will release like this. So releasing in the sense contact sites, once they release, it becomes again like this. Then finally the recycling of membrane receptor from the rear. So here it will from the rear, recycling of the membrane receptor from the rear, from the back to the front of the wall. So this moves like this. Understanding? It's like goes like this, attaches like this, cytoplasm contracts, it moves like this, and then it goes and rear and releases like this. The cell will start migrating like this. I hope you are seeing and I hope you are understanding. Okay, that and the diagram picturization I will explain it. Okay, so extension of the edge, leading edge, the adhesion of the matrix contracts, the contraction of the cytoplasm, a release of the contact sites, and recycling of the membrane receptor from the rear, from the back side. Okay, so that's how it occurs. So here you can see extension. The first step is what? Extension of the leading edge. So here, because of the number of the cells are increasing, differentiating, proliferating, what happens? This extension will be there from the leading edge. The leading edge in the sense this one. Okay, here you can see the arrow mark. Okay, and then adhesion. The second step is adhesion of the matrix contacts. See here the adhesion or attachment of the matrix, or this is the substrate. Okay, so attachment will be there. Next, cytoplasm contraction will be there. Because of this cytoplasm contraction, once there is a contraction, it moves like this. So since this is like this, so once this contracts, the whole cell contracts like this, right? So it will help to move forward. So that becomes the third step. Next, release from the contact sites. Now here, from here there will be a release. Release in the sense from this substrate there will be a release will be there, so that it will be pushed front. Okay, once that happens, the subsequently recycling of membrane receptors from the rear to the front of the cells. Recycling membrane receptors in the sense here release, these are receptors are there, no? The same thing, they will release. Okay, once that is released, the cell will move from this to this. Okay, last two parts are they happens at a single time, the release of the contact sites and recycling of the membrane receptor from the rear to the front of the cell. So that's how the cells are moved, front. Understanding? So see, now the receptor was here and now that it is being moved. Okay, so that's how the cell migration happens in 5 steps. Okay, if it is asked for maximum marks, give introduction, explain these factors, okay, in sentence, okay, like 5 sentences you will get, and explain 2 sentences in every step. Whatever I have explained, that is more than enough to explain this particular thing and include the image. And next also you continue explaining the process of cell movement also, how the cell begins to move in response to the external stimuli to its surrounding environment, right? So here we discussed about the 5 external stimuli, right? Same thing. So this can be a physical, chemical, diffusible or non-diffusible signals that is detected by the receptor proteins, which is located on the cell membrane and transmitted by them via signaling cascades to the cell interior. That means how this cell, how this particular, all these things are getting all the signals for this. So when there is soluble concentration gradient is different, then that soluble the cell outside cell surface proteins like proteins will sense that, and that signal will be given to the particular cells. Okay, so that's how the things works. So they're located onto the cell membrane and transmitted by the via signaling cascades into the cell interior. Now a cell such as white blood cells or yeast cells or slime mold cells is believed to sense the signal direction by the spatial recognizing external gradients. So this WBC yeast cell and slime molds, these all things, they will sense the signal directions which are coming from spatial recognized external gradients, like receptor proteins become more concentrated on the side of the cell where the signal is present. Wherever the signal is present, obviously the receptor proteins will tend to move that side. Okay, so once the cell movement begins, the process which involves in the constant reconstruction of the actin cytoskeleton, that can be divided into 3 stages into most cells. Okay, what are those 3 categories where this actin filament is involving, actin network is involving in division or the movement? The first thing, a cell propels the membrane forward by orienting and reorganizing, reorganizing in the sense here it is growing the actin network at its leading edge. So same thing what we discussed. So this is the protrusion of the leading edge. That means how exactly it is moving. Consider this as a substrate, direction of motion, actin network is there. So a cell propels the membrane forward by orienting and growing the actin network. Once this actin network grows, and then this membrane will be forward at front. Okay. So second, it adheres, again attaches to the substrate at the leading edge and de-adheres, releases at the cell body and rear. So here it moves like this, new actin will come and new movement will be there, new addition will be there. So here 3 things were there, now it went like this. Okay, so in second step what is happening? Addition at the edge, leading edge, that is cortex under tension, and because of the movement, cell movement, the movement of the unpolymerized actin, and here it attaches, and rear, that is de-adhesion at the trailing edge or rear side or back side of the cell. So this will be reattached, so the movement will go front. And finally, contractile forces, or you can say generated largely by the action of the actomyosin network, which will pull the cell forward. So that's how this will go. See, from here, protrusion of the leading edge, which is elongation of the leading edge, then adhesion here, then the release. This adhesion and release is happening in the second step, and movement of the cell body. So now what you understood? So whatever these things he explained in 5 steps is also seen in the process of cell movement. That means it is simplified and explained in terms of the different cells, okay, with respect to the cell signals. Here it is explained generally how exactly the movement, cell migration takes place. Here cell movement, okay, the process of cell movement. So there is no much difference, both are same concepts. So some of the factors are there that are affecting the cell migration speed. That means how fast they are migrating. That is affected by few few factors. What are those? There are two factors are there which are going to affect the speed of the cell migration. The first one is substratum adhesive strength, that is nothing but attachment strength of the cell, okay, when there is a movement is there, because that is the second step usually. And next, intracellular contraction forces, that is the third step where the cytoplasm contracts, right? So intracellular contraction force, how exactly the contraction force is there inside the cells, understanding these two factors. These two factors has to be intermediate. That means the adhesion should not be too much and contraction force should not be too much, understanding. When they both the factors are intermediate, or you can say both the factors are optimum, or not too low or not too high, okay, then cell spread will be significant. This is how the cell spreading can be seen. But imagine the adhesive strength is low but contraction is more, then we will see something like large cell spread, because contraction force is more, so the cell will spread. Whereas adhesive strength is less, though attachment will be slow, so the cell movement will be random cell movement will be there. There is no proper movement because these both factors are not even. When you that was about the large cell spread. And what about the small cell spread? If the adhesive strength is high and intracellular contraction is less, then we will see something like this, because it is attached at one end very forcibly, because adhesion strength is very high. That's why you are getting the uneven the cell movement, cell migration. So in order to have the proper or a significant cell spread, you need to have the substratum adhesive strength should be in cooperation with the intracellular contraction forces, because if the force is too high, if the strength is too adhesive, attachment is too low, that is also a problem. If the force is too low, adhesive is high, that is also a problem. And if both are incorporate with each other, then it's called as intermediate cell spread, which will be significant, or else it will be negligible. So these are the factors affecting the cell migration speed. This separately you can write in sentence. This alone can be asked for 5 marks, understanding. I hope whatever I am explaining here, that is more than enough to understand, because there are only two factors are there. Obviously in one case, large cell spread case, what happens? Adhesive strength, that is attachment strength, is low, whereas contraction force is more. That's why the cell is spreading, because they are not attaching properly. Whereas second thing, what happens? Intermediate, both are same, both are equal, both are not too much too high, so the proper movement is there, that is called as intermediate cell spread. Whereas third case, small cell spread, because here the adhesive strength is high and contraction force is low, that's why the uneven movement of cells or migration of cells. So these are the factors affecting the cell migration. So that's it about this module. I guess we finished for module 2. Next we'll be moving to the module 3 in the next class. If you have any doubts, please write it to my email ID, which will be there in my YouTube channel. In most of the videos you can see. Okay, chalo then, bye, take care.